Alaikum. Good morning, good afternoon, good evening, depending on where you're joining in from. And again, this is the second in series of the research webinars from the Apna Merit, where we are looking at the healthcare research and innovation landscape in Pakistan. We took a hiatus for a few months during the winter time, and then we came back with a new series. My name is Dr. Sarfraz Hasni, and I'm a director of the lupus clinical research at the National Institutes of Health in Bethesda, Maryland, and I'm a rheumatologist by training, and I will be moderating this session. We have a couple of panelists. Our speaker today, the main speaker today, and then we have some other speakers from her team who are joining a little bit later. So Dr. Samina Malik is the pioneer member and chair PG and faculty research at Research Cell of UCMD, University of Lahore. She has introduced the strengths of introducing the strengths of University College of Medicine and Dentistry by presenting eminent research and resources of University of Lahore during this session. She is the head of Department of Physiology, and during her master's in medical education, after qualifying for certificate and diploma from Dundee, UK, she has discovered some novel mutations in breast cancer patients in Pakistan, and has discovered biochemical markers of high specialty and sensitivity as her PhD thesis outcome, and presented her findings on diverse national and international platforms, and conducted a plethora of educational and research workshops. She has a long list of achievements that she has, and I'm sure I can go on and on talking about her. But I think in the interest of time, I would ask Dr. Samina Malik to please start her presentation. Again, thank you so much for taking time out of your busy schedule, and a huge welcome from the Apna Merit Committee. Please take it away.
Thank you so much, Dr. Sarfaraz, and thank you, Dr. Uzair and Apna, the moderator as well. So I would like to start from the message of our principal of University College of Medicine and Dentistry. She could not join in person, but she has sent this message for you. So I'll be playing that.
Bismillahirrahmanirrahim. Assalamu alaikum and a very good evening. Dear students, faculty members, researchers, on behalf of University College of Medicine and Dentistry, University of Lahore, it is my absolute pleasure to be present here on the seminar that is arranged on the clinical research, opportunities and challenges. I'm grateful to the Apna team for giving us a chance to tell us about our brief research landscape at University of Lahore. Well, as all of you know, that research, it plays a crucial role in the advancement of medical sciences, and it is essential to train the next generation of medical professionals in research methodology. With this aim, University of Lahore, they have relaunched the undergraduate research program under the Research Cells umbrella. In this program, we have developed a longitudinal research module for the students that falls under the Pearl module. This module, it spans from first year till fourth year. In addition to providing the theoretical knowledge of research, students, they are assigned research mentors to provide continuous guidance and supervision throughout their research journey. I'm sure that this program, it enables the students to think out of the box, enhance their problem-solving skills, and polish their leadership skills. By the end of the fourth year medical, students, they are expected to complete their research work and develop and submit a manuscript which is addressed by a panel of experts. Basically, the goal at our college is to ensure that our students, they are well equipped to tackle the challenges of the clinical research landscape, and they should be prepared in advance to take this challenge to a new level. We are proud to be at the forefront of providing a comprehensive research program that prepares our students for the future. Once again, I'm grateful to Apna Merit team for giving us a chance to talk about this very important topic. Thank you.
So thank you so much, Apna, for having us. And I would like to add that as you can see that we have already established the culture of research with the help of our dynamic principal and dynamic research chair, Professor Munir Salimi. we would like to tell you that the University of Lahore is blessed with 7 hospitals, and it is one of the largest private sector medical university in which medicine is one of the several disciplines. And it includes the University Teaching Hospital with 406 beds, PESI Social Security Hospital, Multan Road with 400 beds, Al Khidmat Mansoura Teaching Hospital with 170 beds, Social Security Hospital, Kot Lakhpat with 100 beds, Sehat Medical Complex, Khanjarwal, and Sehat Medical Complex, Lake City, and also University of Lahore Dental Hospital. So
I think your slide is not moving.
no, this was not on a slide. This
Okay.
was verbal. So this is the program that we are going to follow today. So I'll be starting from my PhD project outcomes, and it was conducted under the supervision of worthy principal, who is also a PhD in physiology and a master's in medical education, and now she's progressing towards her PhD in medical education. So along with her, Professor Arif Malik, who is my supervisor in this project. And this is a huge project of on breast cancer in Pakistani population. And it included several hormonal and oxidative stress markers, as well as a total of 27 genes that were screened in these patients. So the part one of the thesis, I would say, it included the inflammatory and stress markers and determined that which of them have the highest sensitivity and specificity, so that we can use them in a diagnosis, very early diagnosis of breast cancer in patients who are even the patients who are prone to breast cancer, or those who have a family history of breast cancer. They can be screened using the outcomes that were achieved. And the samples, they were drawn from around 500 cases and 500 controls. And they were tested on several oxidative stress markers and antioxidants. So this is the total of serological markers that were tested in them. And the results were presented, this part of the thesis, its results were presented in Philadelphia in experimental biology meeting. So this was the poster view of the results. And the abstract was published in the FASIB journal. And in summary, we noticed that the oxidative stress markers, their overbalance and underbalance of certain antioxidants, it is responsible for breast cancer in our population. And the main highest specificity and highest sensitivity on ROC analysis was found to be in heat shock protein 27, matrix metalloproteinase 9, and interleukin 1. So these three have been identified as the early markers that can be determined in their blood. And the mechanism is under construction, the genetic mechanism, and this is the other mechanism that might be responsible for pathophysiology of breast cancer in our population. As you can see, this diagrammatic view of how the breast cancer might be caused in our population. Then the second part of the thesis, it included 27 gene panel. And this gene panel was the biggest that is used in Pakistan. And it was used in the form of next generation sequencing, which was confirmed on Sanger sequencing, obviously. And again, we also included the in these patients with the metastatic breast cancer. We also checked for the markers like ER, PR, and HER2, the receptors. These were the genes that were used in two recent studies. So these 15 genes were also included. And this was the total panel of 27 genes that was screened on the patients. The purpose of this part of the study, or the genetic part was the study, was to investigate if there is any interrelationship between the modifiable and non-modifiable prognostic factors and oxidative stress in these females. And the outcome of it also showed a number of novel mutations that are novel, and they are only present in the Pakistani population. So this Pakistani population can be screened on these mutations as well. Within these genes that were screened. So not only that, but their unaffected sisters of these patients, as well as other normal population, because the incidence of breast cancer is 1 in 9. So it is an alarming incidence. So we need to have some data. And this is the real data that we have obtained. And the Breast Cancer Research Center will follow, inshallah, at University of Lahore, which has been approved by the chairman, Dr. Awas. We are going to establish not only a database, but also a research center and a counseling center for these patients with breast cancer, inshallah. So the application of the outcome or the mutations, variants, it would be that we can design a probe, and that probe is able to pick up all these variants that have been identified in this population, and it will be much cheaper than the one that is borrowed from USA. So different epidemiological factors were also determined, as you can see. All these factors have been ruled out, and these factors as well. And these clinical investigations, they were conducted. Obviously, the breast cancer was diagnosed on histopathology. Complete blood counts were also checked so that we can identify that which changes are seen as early markers. So as you can see that with the help of this model, a pathophysiological model, we can design the different drugs, and we are at the stage of drug designing now. And later on, after designing those drugs, they can be used into clinical trials so that they can be utilized in the long run on patients. So with this, I would like to end my talk and hand it over to Dr. Ghazala Ruby, if she has joined. Thank you so much. Dr. Ghazala, have you please joined? And the question and answers, they will be taken up at the end. So do we have Dr. Ghazala Ruby?
Yes, please.
Yep, madam, please go ahead and introduce yourself.
Okay. Bismillahirrahmanirrahim. Thank you very much for inviting. That's a good chance here. I really want to thanks to Apna and all the platform of the UCMD. Actually, I'm working as the Director of Research at Lahore General Hospital and running more than 40 research projects, in which 4 are the international projects and 2 are the clinical trials as well, in which I am the PI as well on all these projects. So let's start with the presentation.
Excuse me, madam, can you open your camera?
Yeah, please.
Thank you so much.
डॉक्टर रूबी, आपको मैं एज ए पैनलिस्ट इनवाइट कर रहा हूँ, आपको ज्वाइन करना पड़ेगा।
जी, ओके।
थैंक यू।
Okay. Is it okay to everyone?
Yes, we can see your presentation. Please go ahead.
Okay. Let's start with it. Randomized controlled trial, that is RCT actually. The aim of this RCT is always to test some medicine before applying to the community and let it get public. So the main procedure should be opted to use it as the clinical trial or the randomized controlled trial in the society, so that they become the clear scientific evidence to eliminate every form of the causality and special condition of the randomized controlled trial. But there are some ethics, biological ethics, social ethics, and the technical ethics to run these trials. So the basic idea is that the new treatment should be allocated to the subjects, to the patient at the random of the different treatment level. They should be statistically equivalent to all of the level. Clinical trial is sometimes disease treatment, whether we want to continue or the yes or no. It means that, for example, we want to introduce the placebo population random selection in the clinical trial, and the investigator apply the treatment and observe whether the outcome that are coming, they are effective, they are effectively acting on the patient, and patient are getting treatment very well done and getting cure without any harm effect to the patient. And the main advantage of these trials are that to conduct the observational studies in the area where the ability to demonstrate the population, and the randomly assigning the interventions so it can eliminate the influence of confounding variables and blind the administration so that it can decrease as up to the certain level. Next, please. A randomized controlled trial, there are different steps in it, that the selection of the participant should be very careful. When we design such a type of the trials, we have to approve all ethical aspects of the trials, that the participant, whether the drug is for the adult, and it should be included from 18 year to 50 year or 60 year, and what kind of the patient or the people can participate in it, whether the healthy non-pregnant female and the other patient who are not having the comorbidities, and random allocation to the treatment, and then blinding of the treatment or control of the treatment. But we have to help with the clinician as well, because clinicians are going to apply these drugs or these treatment to those patients. For example, sometimes we are not only doing the clinical trial for the drugs or the treatment. This trial, which we have approved recently in the March from the DRAP, that is actually the device which approved. Device for the treatment that we have got approved from the DRAP. So it means that these things should be first applicable to the patient at a control area, and then we can find out, then we can exclude whether this treatment or these experiments went very well, so that we can apply it further in the society in a very approved manner, and then we can patent as well. So the sample size calculation, that is very important, whether they should be at the large scale or the small scale, at the random minimal scale, and the area of allocation is very important, that the population on which we want to conduct this study, they should be having from the different geographical area, not from one, but they should include some patient from the Asia, Africa, and then Europe, and then America. So it means that we have to include all those samples from the different variables from different geographical distribution, because we have to apply this drug to all the areas. For example, if some drug is going to introduce in Pakistan, and those trials have been done in foreign, in US or Canada or UK or Australia, then we have to do these trials in this population, our population, to conclude whether they are effective to us in our population as well as they were effective abroad. So it means that we have to compare these drugs with the other people as well, or the other studies, but we have not rule out these study that this have been approved over there, so we don't need to be approved over here. So it means that these studies should be done in the area, and they are going on as well. So the sample size calculation is important by the formula or by looking on the geographical distribution as well. And then inclusion and exclusion criteria, which I recently told you, that the age group, the patient type, and the people who are really want to take part in the clinical trial. Because the other disadvantage, because I'm running a big clinical trial with the help of the UHS, and the people are very hesitant to take part in the clinical trial. You have always to get them convinced that it will not harm you. So you have to, counseling, it is very important in this. You have to properly counsel the patient. Even many of the doctors of our hospital, they are not interested to take part in the clinical trial. So the informed consent is important in that case, that if someone is not interested and the sample of that patient is with you, but if he or she is not interested, you cannot use the sample for the study, for the trial, until and unless he or she is fully agreed to take part in this research. So it means that the even distribution of these factors, they are very important. Next, please. So by opting the new treatment without conducting the trial is not a good idea. So we have to always consider until and unless the trial became successful, you cannot start those treatment. You cannot start those drugs. You cannot start those devices. You cannot start those diagnostic tests. So it means that continuation of the trial, if the treatment is harmful, you have to discontinue, because this is non-ethical. So what if the treatment effective? Yes, if the treatment is effective, it means that your trial are very good, and you have conduct the trial with the marginal benefit. There are a lot of advantages and the disadvantages of such kind of the trials, that we demonstrate casualties when we do the randomization, and sometimes we eliminate the influence of confining variables. But there are disadvantages as well, that expansive compliance, when we select the population, there are the ethical issue. I just described those ethical issues, that many females, they are not interested until and unless they get approval from their family whether they want to take part in the clinical trial or not, because they don't want to get blamed that after the trial they got such harm. If not getting harm as well, they must correlate it with as well after that. So it means that we have to carefully select the subject in these clinical trial. This is not the normal research study. This is the clinical trial where you have to inject some medicine or treatment or some device or some diagnostic testing. So it means that application of the drugs, sometimes people think that they are going to be used as the guinea pigs, so they should not take part in such a type of the study. While I have spent a lot of time in US and the UK as well for the research, and they are very happily want to join all these clinical trials and then their such kind of the research. Because one of the trials which we are conducting, at the same time, it is going to happen in UK and Australia, which is also we are doing in Pakistan and Lahore as well. So while we conducting the clinical trial, we have to first take approval from our local ethical committee, approval from the institute, that is the IBR. And then other one, the important one, that is we have to take approval from the National Bioethics Committee, and the final approval from the DRAP. And then you can easily get tested and approved. And after conducting the successful trial, after the DRAB approval, you can get patent as well by getting on register on the website. So these shunt patients should be, previous slide, please. The study population is very important. So the sample size, some group 1 and group 2, and the variable, what kind of the variable you have selected with this one. What is the infection type which you are going to treat with these drugs? Antibiotics, if the patient is taking any other antibiotic, you cannot take such kind of the subject or the patient in your study. So the dummy table and infection, yes or not. Next, please. We have to critically think that what is the outcome of the study and what kind of infection you are going to the treat. And sometime if the patient get complain after half of the trial, after the few doses of the trial, that he is getting complain or he is getting sweating or asthmatic problem or something like that. Because recently we have advised the nasal spray for which the people sometimes complain that they got asthmatic and they are heaviness or something like that. So you have to stop those trials from those patients, and you have to exclude those patients from your studies, and you have to stop the trial on those patients rather than infecting them more. Or it is not the idea that you put some other infection to the patient rather than having the treating them in a very well manner. So the total therapy is concluded that which kind of the therapy was inducted. So it means that the target therapy, main aim of the clinical trial is that you have to achieve the target therapy to treat the patient in a very well manner, the specific therapy. So it means that there is sometimes relative risk and you have to reduce the risk by taking part in them and reducing those patients as well. So whenever we are trying such a type of the clinical trial, statistically approval when your p-value is less than 0.005 or confidence interval is 95%. When the value is significant, it means that the results are very good and these trials are successful. You can apply it to other areas as well. Yes, please. This is our approval that we got it from the DRAB, and this drug is along with the device as well. So our device plus drug has been approved from the DRAB, and so we are going to register it very soon, inshallah. Anything else, please? Thank you very much from my part.
Thank you very much, madam. Question and answers will come in the end. Please stay tuned. Thank you so much. So next we have in line, and I would request the admin to kindly make Dr. Atif Amin Beg a panelist as well. He's the Deputy Director, International Linkages Cell at University of Lahore. Welcome, sir. Please go ahead.
Assalamu alaikum warahmatullahi wabarakatuh. Thank you so much, Prof Samina, for introducing me. And also very thankful to the respective APNA committee and all the members here who are actually giving their bright time and ideas to us, inshallah, in the future and also now. So my name is Dr. Atif Amin Beg. I'm currently heading the international linkage cell for the University of Lahore. So without further delay, I would like to just continue my slides. The first of the important part here is actually I've been back to Pakistan approximately after 23 years. And I found interestingly, the big issue is that 5% of that statistics, which we have to be worried about, just we have the research methodology, then we got the bioethics, then we have the biostatistics, and the last part, the research write-up. But the majority of the people, especially the students here, I found they are so much curious about the research statistics. And for that reason, that is a fear when they wanted to start, they got stuck because of the research statistic part. So we are trying to have it easy, and we are trying to make them relax now with my presence and some other people here. So the first part, we have to overcome the statistics part, that 5%, we have to let them know that that 5% is not that important. Important is to target the rest of the 95% of the research. Okay, now we have to educate them a lot for that, and we have to talk to them on a personal basis. And we have lot of the researchers who are into economical revenues, doing the analysis for money, and it is really affecting altogether our research part, especially in Pakistan. Okay, now we are looking at the quality of our research currently, because we have the primary sources and the secondary sources. In this triangle, you can see in the hierarchy, you can see down there, we have till the randomized controlled trial is all in the primary sources, and up there is a secondary sources. And the topmost is a systematic review, and above that we got the meta-analysis. And then yes, before sure we have the synopsis, thesis, and policies with evidences. So all those way is already there, which we really need to discuss. But the most important part I would like to highlight with all the respective and senior members here who can make a difference basically. Interestingly, we don't have even a single vital registry in Pakistan. And looking at the data, when we talk about the big data sciences, we have to look at the John Hopkins, which are normally taking the data usually from some of our offices or at the Prime Minister office, and we have no access to a lot of our data, which we have to, we need for a lot of big data sciences. Or either we have the option of very interesting the GBDs, the global burden of disease, where we can get some vital data. And interestingly, luckily we have GBD where we can access the data. So this is another issue. And what we are targeting here is, because randomized control trial has been talked very nicely by Dr. Ruby. So I'll just highlight some other few issues which are very important for especially the students who are attending here. So moving on to the next slide. These are the normally when we talk about different kind of study design which you normally face. We have the experimental under which we actually target in most of the RCTs. Then we have the observational, we have the descriptive. When we talk about the observational cohort, we end up in the relative ratios, and when we talk about the other part, we got the odds ratio. So this is just a summary for the youngsters here. Can we move on, please, to the next slide? Okay. So what we are heading, I have been a part. I was actually requested to attend few of the ethical review committee boards, and interestingly, what I found, majority of our data, they are targeting basically not the community trials, but basically it's a very small sample size kind of data, which is focusing on a very small minor kind of studies, claiming for sure it's a RCT. But when we look at the statistical rigors, it's like really undoable in the real life scenario when we really want to use it. So the difference is community trials, we have very, very minimum. The reason could be the way they claim, saying it's because of the cost of effectiveness. It's caused because of a lot of samples issues. And the most biggest claim is because the students, they wanted to graduate in time, and for that reason, in 9 to 10 months of that candidature cannot finish the entire trial. So the sample size is normally very low. So we cannot use that actually to come up with the proper registries and proper top, even for the autopsies later, if any hospital could align. So what we have is the differences in terms of outcomes, unit of measurements, question answered, and major objective. When we look at the major objective, we have the assess factors which affect implementation versus the clinical trials. We are more into the causal mechanisms. So can we move on? I just was to save the time. So okay, this is in case of cohort study, some of the students are involved. So normally we look at the relative risk, and the relative risk is very important when we are targeting a study at a bigger level. Please, next slide. And this is another, just a summary. When we look at the case control kind of studies, we look at the odds ratio. The direction inquiry is opposite to the cohort study usually. Thank you so much. Next slide, please. Okay, this thing is very important. I just want to highlight, you can actually access the clinical trials website. And the reason I brought in this specific website is because interestingly, we are in an era where we follow the pragmatism. Pragmatism is to follow the criteria. If you know the rule of the game, you win the game. If you don't know the rule of the game, you lose the game. So currently, I assume looking at the clinical trials at NIH could be the best option, because I've been last time working with them for a while. So I found better to share this form in which you have a better idea. So can you please click? There will be some, yeah. So we have here, if you look at the study type of studies, we have the intervention, we have the observational patient registry, and the expanded assess. So if you look at 4 of these kind of studies, make sure, my suggestion, we need to align back our all the ethical review boards to this form basically. I think majority of the stuff which is needed has already been covered in this form. So this is the section 1, which is a section 1 of this form. It's a study identification. So normally if we target interventional, then we have to move on to the next levels. Thank you so much. Please, can we go to the next slide? Okay, so second one is the study status. So here you put your everything, the related relevant information. The most important part is if you look at the overall recruitment status, sometimes you've already started your study and then you want to still register at the clinical trial. Now the question is, why you really want to register at the clinical trials? So the most important part is because if someone is really interested to see what is happening in his country, or if you want to do something new, like what Dr. Ruby said, you just go to the clinicaltrials.gov. You can select your city, you can select your country name, and you can just Google it out. You can just search it out, like what are the current trials going on in your country. So maybe you are repeating something, or maybe something is already there, and you can actually go to the next extended part of the study. So you couldn't repeat, and you will be more useful for your country and for your place. Interestingly, what is happening, what I found, especially to the respective APNA committee here, after coming back, I interestingly found the people here, they love to collaborate with overseas. But in collaborating, usually what we have been into, we collaborate in terms of solving the problems of other countries. HGC is very prompt in, the international collaborations, but we, instead of looking into local solutions, solving the local problems, we are actually into solving the global problems. So I'm more into saying, like, we have to solve the local problem with a global impact that could be more better for us. So for example, maybe Pakistan main issue example is Mycobacterium tuberculosis, rather than going to something leptospirosis, better we focus on that part first. So that kind of is still missing here. So okay, now we go to the number 4, this oversight. So this is focusing, like, is all this relevant information in US or not? Can we go to the next slide? I think we're running out of the time, so it's getting forward. Okay, study description, we have to add in whatever we got in our proposal. We have the conditions and keywords. Can we move on? Okay, it's the study design is very, very important. So you have the you have to click the proper relevant information, which is in alliance with your basic proposal, which you have been into, which you've written before. So you have to be very clear about it. So you have to read everything, what is phase 1, phase 2, phase 3. You have to be very clear about all of these things. Move on. Okay, so this is something about, like, your grouping of, like where, how many groups you have, how many AMs you have, how many clusters you have. So everything is you have to add in the section 8, and especially one of the terms is the sham comparators. You have to be very sure what are those. I've already mentioned them here. Next slide, please. Okay, so these are the outcome measures. You have to be very clear about your outcome measures, and you just use your actual proposal, which you have brought your REC or your ethical committee. Use it specially to align here. Next one. Okay, now eligibility, you put your eligibility criteria, which is more like inclusion exclusion criteria. You have to add everything here, and especially please align it back with actual REC or ethical board, which has cleared your application at your institution. If they are not aligned, your application will be sent back and you have to revise everything back again. Thank you so much. Next one. Okay, so this is, you will add your all the context, all the details of the PI, especially the sponsors. One, we have the sponsors. One we do have the people who are not the PI, but they hire some other person. For example, you have the PI or the molecular geneticist, but the study is, the samples are being collected by basically a surgeon. So that person name must be included in a proper way. If it is not there in the REC record at your institution versus in the clinical trial, then you have to revise everything again. Next one. Okay, so this is something very, very important. It's optional for basically clinical trials, but it is very important for ICMGE. And this is IPD, which basically stand for individual participation data sheet, or IPD sharing statement we have to add in. So in ICMGE, that is International Committee for Medical Journal Editor, when you're publishing your data later, it will be a very big issue. So this is altogether, I think, 12 of these stuff. Move on. Okay, so this one you have to add in all your details of IPD, then followed by the references. So what are the references you're adding, your citations, everything you have to add in for your specific, the project you're uploading. Move on, next one, please. Okay, so this one I'll just move on to some of our current work with purpose collaboration. This is one of my team's current project in which we are targeting the acquired immune response in case of Streptococcus pneumoniae using netosis. What is netosis? Netosis is basically coming from NATS, that is neutrophil extracellular trap. So the new invention is suggesting like the nucleus of the neutrophil, it got protruded and it will come out and it will try to capture the different bacteria and then present it back to the neutrophil, and there are different mechanisms by which netosis happen. So this is these new nets are made of the chromatin plus protein molecules. So what we finally found a mechanism in which we are able to basically get the capsular Streptococcus pneumoniae, and it can be taken up by the netosis. But currently my PhD student is working on the animal models, and we are going to move forward next to the clinical trials, inshallah, after this phase. So this is one of the project we'd like, if anybody was interested to collaborate, we can work together. Next one, please. Okay, this is one of the study which was conducted in Pakistan, but there are few of the people which are not in Pakistan. It was basically parallel study design. It was both randomized controlled trials, and I think they're already in process, they're ongoing. So these are two other studies which were performed in Pakistan, but again, it's a parallel study design, one of the examples. We have currently doing two other projects, which I currently did not add it here because of time constraint, but if somebody is interested, we can share, inshallah. Next one, please. Okay, this is one of our study in which it was under the prophetic medicine. We are currently working on it. So what we did, we took the promagnate, which is basically anar jise aap kehte hain. So we actually pick up all bioactive molecules. From then we actually use that the bioactive molecules against the opioid dependent mu opioid receptors, and basically we control the dependencies and all those kind of the what we call all those kind of dependencies versus tolerance kind of thing in opioid patients, opioid subjects. And from there, we come up with one of our specific, what we call the product, which is currently in use for this non-opioid therapy for the opioid treatment. Next, move on. Okay, this is one of our current recent projects. We are looking at the association of genetic genes with obesity in Pakistan. Secondly, our main interest is role of hypoxia and hypoxic genes in obesity in Pakistan, inflammatory genes in obesity, because currently the new data shows and the new studies shows that obesity is because of the inflammation and hypoxia. Remember, the hypoxia basically triggered NF-κB and TNF-alpha, and this is what mechanism happened in the adipocytes. So where we are heading now is we are looking in our population that our obesity is basically a metabolic kind, it's the inflammatory kind, it's a hypoxic kind. So we are into that kind of strategies, and we are screening more than 14 genes currently with approximately 5 masters and PhD students, and we are looking forward for more genes, inshallah, in the future as per our resources, inshallah. So then we have the anti-obesity strategy for obesity at the molecular level, in which we have found that one of the way we can reverse the cellular hypoxia, and the article is recently accepted in Nature, and inshallah, I think we will be sharing it soon. And the last one is anti-obesity drink targeting specific genes. We use a lot of money to make that drink, but we have a small issue with one of our group. So we got stuck, and we are just revising some of that data. Inshallah, once it will be done, it will be in the market. We have an app is called Obesity Socials, which work on identifying the social factors which the obese person is having, and if you exclude those factors, there is a chance of getting rid of obesity, maybe at 30 to 40%. So this is one of the first app which is targeting the reducing the obesity and the social factors in obesity. And now currently one of my PhD student is using deep learning and artificial intelligence to target the globesity, like the factors of obesity globally. Next slide, please. Okay, now this is one of the things we are into currently, and we are working out to apply for a few grants, because spinal muscular atrophy is one of the neuromuscular disorders, and its incidence is 1 in 10,000 live births. The Roche in Pakistan, interestingly for the APNA team, the Roche Pakistan was unable to get a chance to promote the trials of the new drug in Pakistan. So currently Indian Roche is the one which is taking a lead, and Pakistani patients of SMA have to register with Indian SMA to do all those kind of trials, but it's too super costly. It's nearly 100,000 in Pakistan for one subject. So we are into more looking into now nutrigenetics and nutrigenomic kind of approach, target the histone acetylators kind of thing. Maybe it will work. So currently we are into analyzing the core promoter and promoter regions of the genes to target the transcription control of these genes. So if anybody would like to collaborate, will love. The last one, I think, is the role of genetics, like actual assessment scores. I feel my students working with the medical education, and we found, like, we assume that maybe there is a chance if we that metacognition is under the control of the genes, and if we control the metacognition, there is a chance we can control the actual assessment scores. Similarly, it goes to empathy, the same thing. That, and we are getting a positive results for that. For example, recently we found the brain-derived neurotrophic factor, the mutations in brain-derived neurotrophic factor is the reason of the stress in majority of our preclinical students, in nearly 60% of the students. So it is highly associated with that one specific mutation. So what it means, we are not saying that our future doctors are not going to be the good one, but we can control the mental system. Okay, now this is in process development of DNA vaccine for cholera. If somebody would like to collaborate, move on. Okay, so this is something, the phenotypes of Streptococcus pneumoniae in Pakistani population. The idea is there is only 2 phenotypes, and under these phenotypes, there are different antibiotic markers. So my point is, like, instead of targeting the antibiotic markers, going for the culture sensitivities take long time. So why not we just target either the phenotype or the PCR based through the phenotype to just diagnose our phenotypes, and we start treating the Streptococcus pneumoniae. Next one. Okay, this is one of our global project. I would just like to congratulate and thank you, Dr. Samina, for helping me out in this project. This was under the Human Variome project. I've been working nearly 13 years as a data manager for them. And this Globin network was basically proposed by me nearly 2 years, 1.5 years back, basically. And we come up with a new criteria of increasing the quality of the healthcare system in hemoglobin pathways. So recently, Alhamdulillah, we happened to bring the Pakistan Global Gene Network to Pakistan, and we are planning quite extensive projects. Currently, we have been offered for nearly 180 samples for next generation sequencing in Italy, and we are going to send samples from like approximately 30 to 40 samples from every state in Pakistan. We are planning for our own database. So I hope I would like to collaborate, and we can have to do something good for Pakistan together. So these are kind of collaborations, University of Lahore, and we would like to offer. And if we are considered to be, to align with others, all of you, the first one is the joint research projects, joint publication, books, grant application, student supervision, mentorship program, which could be for distance learning kind of stuff. We have the student exchange and transfer programs, MOA, MOUs, and then invention and innovation. Interestingly, I would just like to highlight, as per our symposium, because we have the idea generation step number 1, followed by the prototype for invention, and third one is the innovation. So in Pakistan, currently we accept the DICE. There's one of the program DICE which are working on the innovations, but majority of the cases, we are actually at a level of idea generation, and the idea generation is without evidence. So this is one of the culture we have to bring, and maybe we can do it through APNA in next few seminars, in which we can target the, bringing the idea of what is invention and innovation. Thank you so much. I have to rush again. I'm so sorry. I take extra 2 minutes. Thank you so much. Thank you, Dr. Samina, and thank you,
Thank you
team.
so
Thank
much.
you so much.
Thank you so much, Dr. Atif. And we have now the co-chair of Research Cell, Professor Dr. Saima Batul. She is head of Department of Peds at University College of Medicine and Dentistry. She'll be introducing the Research Cell, please. Yes, Dr. Saima. Assalamu alaikum. Please go ahead.
Assalamu alaikum, everyone. I'm Professor Dr. Saima Batul, and I'm working as Professor
yes,
of
Dr. Saima.
Pediatrics at co-chairperson. Can you hear me?
Yes, please. Go ahead.
Okay. I have a brief presentation about the introduction of Research Cell of University College of Medicine and Dentistry, the University of Lahore. Basically, the research cell was developed almost 5-6 years back. We have developed a vision and mission for our research cell. The vision being to promote quality, innovative, ethical, and evidence-based research for advancement of knowledge and healthcare. Our mission is to achieve excellence in creditable research by creating conducive environment of learning, training, and provision of all necessary resources. These are the terms of references and standard operating procedures. I would go into the details, just a brief depiction. The composition of this research cell, the patron chief, which is our principal, Professor Dr. Mahavash, then the chairperson, Professor Dr. Munir Akhtar Salimi. Next one is the co-chair, which is myself. And under us, there are 5 different committees which are working. These, the chair and the co-chair, they are basically nominated by the competent authority of UCMD. And the rest of the people, they are further nominated with the consensus of the chair and co-chair. These committees, they are basically nominated for a period of 2 years, and they also include the students of both undergrad, at least one student from the research club, and another one from the postgrad students of medicine and surgery and allied. So this is the hierarchy. We have 5 committees. One is related to the faculty and postgraduate research, which is headed by Professor Samina Malik. Then an undergraduate committee, which is headed by Professor Ghazala Irshad. She is head of biochemistry. The third one is basically related to our journal, although it's fancy, not very old, only 2 years back. It was rejuvenated, I should say, and it is headed. The main person who looks after it is Professor Maria Hedayat. She is the managing editor, professor of anatomy. Then we have a research club, including the students from first to final year. The final year being the senior most class, so the president is chosen from the final year, and then the vice president, general secretary, internal and external secretaries from the lower classes, an ethical review board, which basically is responsible for looking after all the different types of studies, protocols, or synopses which are being submitted to the research cell. And then after their ERC, it is approved. This is the research cell, which was inaugurated 2 years back by the principal. And you can see Professor Munir Akhtar Salimi, our chairperson, on the back. Meetings we conduct is almost every month. So we try to do it physically, but sometimes we have to go for a Zoom meeting as well. And the meeting, in the meeting, we also look forward for we see that whether the previous, all the things which we have, they have been completed, all the assignments and new assignments being given to the different participants, and minutes are being kept regularly, and 50% of the members, they are present, and the program is complete, and we proceed with the meeting. So the way forward, or what is we are looking national and international collaboration. I am very thankful to the Apna Merit team in which we are going to have a good relationship in the future, inshallah. Then we are also going for different, as Dr. Atif Amin has said, that the various trials are also already being going through. We are going to move towards further clinical trials with the involvement of faculty as well as from people from outside. And inshallah, this would be a fruitful collaboration. Thank you very much.
Thank you so much,
Over to Dr. Samina.
Thank you so much, Professor Saima. Now we have Professor Munir Salimi waiting for the vote of thanks, followed by question and answer session. Professor Salimi, can we please have you? Thank you so much.
Yeah. Bismillah ar-Rahman ar-Rahim. It's a great honor for me to thank Worthy Chair, Professor Samina, moderator, Sayyid Uzair Saab. I'm really grateful to Apna Merit to really have this particular seminar or webinar, focused on University of Lahore. So we presented how we are working, and I'm really grateful to speaker Professor Samina Malik and Dr. Ghazala Atif Amin and Dr. Saima. But I can tell you it's a really great occasion to have the participant from all over the world. And for me, it's a really great honor to welcome and thank Professor Zulfikar Bhutta, because I think I have all those memories when I was inducted in as a selected member of biomedical on National Bioethical Committee, being a mentee of Professor Famida Jalil. And I'm at the same time also grateful to all of the worthy participants from, for listening to this particular seminar. But I will not hold this much, because I think as the time is short, I think that we should proceed to question and answer so that we can make best use of the speakers, and of course we can have an interaction. So thank you very much, Apna Merit, for really convening and providing us opportunity to present how we are working at University of Lahore. I can tell you from our chairman and rector, and of course all functionaries, research is a great priority for us. And I think in a way we are developing this sort of collaboration, and of course projecting ourselves and coming for collaboration. And that's a really great occasion which has been provided to us. Thank you very much, Apna Merit. Stay blessed. Thank you.
Thank you so much, Professor Munir Salimi. Now over to Dr. Sarfaraz and Dr. Uzair, the moderator and panelists, for question answer session. Thank you.
Okay. Thank you so much, Dr. Samina Malik and rest of your team for an excellent presentation. And I see there are some questions and there's a lot of things in the chat also. I think in the interest of time, I will hand it over to Dr. Uzair, who's our panelist, to take it away and close the session, please.
Thank you very much, Dr. Sarfaraz. So congratulations, Dr. Samina and the team. I think it's a great presentation, very well organized and very efficient in terms of time, selection, and all the criteria. We have just few questions, but then we have some comments as well that I will come back after these three questions. So the first question is from someone. Why don't we use our own data, having clinical trial unit established locally? So this is more of a quite an open question, that why the local data is not being manipulated and used for drug development and just be part of the regulatory intervention and approval. So anyone from your side who would like to respond to this question? Unmute yourself and please respond from Dr. Samina, your team.
Yeah, I would like to respond that just like I mentioned in my talk, that we have gathered a huge data on mutations on our own population. And the prime purpose would be to benefit our own population that is having the incidence of 1 in 9 breast cancer, just to give an example. And this is the panel that we were talking about. This is the Breast Cancer Research Center that we have been talking about, that we will be screening our unaffected sisters of those patients and the other members of the society who are in the reproductive age, the premenopausal women, so that they can be early diagnosis as early as the age of 18 years as per international recommendations on this screening methodology, blood screening methodology, with the help of these mutations, as well as the highly specific and highly sensitive markers. So we will be doing it on our own population, and we'll be gathering the data for entire Pakistan, inshallah.
Excellent. So the next, yes, somebody would like.
Prof,
yes.
can I just add something?
Yes, please.
Yes, Prof. Actually, as per my understanding, one other big issue after I was in Pakistan, that normally we follow the gathers guidelines. That is basically I think, if I'm not mistaken, we called it the guidelines for accurate and transparent health estimates reporting. So for using the gathers guideline in Pakistan, with current system, we are not aligning anywhere. Number one. And secondly, I found in Pakistan, majority of the research, the data collection, either is academic driven because people they really want to do PhD and masters. The option number two is industry driven because industry want, like especially the food industry, आटा चाहिए, नया आटा की type चाहिए, नई whey proteins चाहिए. We are into those kind of research. So the stuff which is we are into now, like with the PGGN, Pakistan Global Gene Network, we are trying to come in with the new vital registries. Can you imagine, like we have 75 years, we don't even have a single vital registry, prof. And the data which is available, it is with like HIV centers, they have their own medical records, but they're all in the paperwork, but they're never been translated into soft copies. So I think we should come up with some projects altogether. We have to bring that culture in which we bring our master's and PhD student, and we just give them projects to curate that data back into the soft form, and we analyze those kind of data. So wo kahin na kahin we start aligning somewhere to be useful. Otherwise, actually, we're wasting a lot of data from like simple thing, pharmacovigilance studies, pharmacogenetics, genomic studies. We have nothing basically to present even to the pharmaceutical companies. So why are they going to, market the stuff here, even very, very cheap stuff, very cheap drugs we cannot bring into Pakistan because of those criteria that the gatherers are missing actually. So I think we should bring those kind of things slowly. And I think that some of the people I know, they start working on it. And I hope we'll be more positive, inshallah, in the future. Let's see, inshallah.
Thank you very much.
I
Dr.
just want
Atif
to
Beg.
add a single comment that I presented the clinical trial in which I quoted that the original one we just approved our clinical trial from the DRAB as well from the UCMD platform. So I described my own data. So that was not somebody else, or I didn't quote the data from some abroad or some other country. I did quoted only the data which we did work on it. That's it.
Thank you, Dr. Ghazala. Excellent. Just to add to the discussion, I would just like to add something that I had a meeting with the Chief Executive Officer DRAB and the Director Pharmacy Services. So there is going to be a lot of work from the regulator, and soon you will see some of the new recommendation, new addition, and a new draft which is in the pipeline. But my feeling is now the team is quite organized. There is going to be a unified body, and Dr. Samina is also part of that, and they are working on putting the recommendation. We will come back to you based on that information. So that's really good, because way back when I was member expert on biological committee with the DRAB, at that time it was known as the Drug Control Organization. So I proposed that all the biological which are formulated in Pakistan, they should have the local data, local clinical research. So that was approved, but unfortunately it was not implemented. Another quick point, there is a R&D fund collection by the Drug Regulatory Authority, which is quite substantial. I believe it's like 2 to 3%. And it is lying over there, and it was never used for anything, and no one applied for that. So that is just, from the sideline. Moving on to the next question, that is from Rahman Institute of Medical Sciences, that they have a clinical trial unit. And their question is, how can we be part of this group? I mean, your group, and how can we be useful? So that is quite open. And this platform is going to give you an access to each other information. And in that way, we are going to collect all the information. And the same question is from the Rahman Institute of Medical Sciences. So they are showing very strong interest to be part of your work. But I'm very honest and frank with you. I'm really amazed by myself. I never knew that so much work is going on in, with this university college in the private sector. That is really amazing. And I congratulate to all of you. So from the question answer session, there is not much. And we have with us Professor Sarwat Hussain. He's a veteran clinical researcher from US, and he is a radiologist. May I request Professor Sarwat Hussain to say a few words? Unmute Professor Sarwat and say please few words, if you're around. It appears that Professor Sarwat Hussain is not around. Now, may I request Professor Umar Hayat? Fortunately, he's in Pakistan to say a few words about his interest to bring clinical research to Pakistan. Over to you, Umar.
Thank you very much, Dr. Zara. I really appreciate it. The presentation was amazing. And I'm really impressed at what you guys are doing, especially with the oxidant and antioxidant factors, and also looking into the genome to diagnose the cancer early on. One question I would like to make: are you working with some pharmaceutical chemistry groups who will be designing those molecules, and then you guys can screen those molecules?
Sir, I'm working with right now with some bioinformatics people at ComSats, and with the help of them, it is bioinformatically we are designing. You mean by computer? What I mean to say, we are designing those drugs. And once computational designing is done, then we will be the next step would be randomized control trial. So then those drugs will be tested on volunteers who would like them to be tested, who will consent, and obviously with ethical approval and all.
Yeah, no, very smart, Dr. Smith. I really appreciate it. I would like to add what Dr. Uzair mentioned. Really, I have a very recent experience with the draft. I was really amazed, like how the things are moving on, and the things were getting done in time. And that was really my personal experience, because we got inspired by this platform and decided to set up a company by the name of Trial 360. And now I will say we are officially CRO, and we would be bringing a lot of clinical trials to Pakistan. So Dr. Uzair, yes, we got yesterday the license, and then we will be approaching to the different companies where I have the network and connections, and we will be bringing a lot of clinical trials to Pakistan, and we would be looking for the principal investigators. We would be identifying the hospitals who can serve as the clinical sites, and then of course we would be looking for the staff as well. There would be a lot of collaborations with the universities, because we are also very active in the animal studies, especially setting up the models for various diseases. Thank you very much, and back to you, Dr. Uzair.
Thank you. We have with us Professor Sarwat Hussain, who just recently joined. Professor Sarwat, would you like to say a few words, please?
Yes, sir. I've actually been listening throughout the presentations, or most of it, and really looking at the 20,000 foot view of the landscape of research in Pakistan. It is extremely, extremely heartening to see that, you know, the research terminology is now rolling off the tongues of our colleagues and researchers, which were kind of difficult and foreign to all of us. So I think this is an indication of tremendous progress, and I think the Sehra goes to this group, and especially the leaders of this group, Uzair Bhai and others. I have this bad habit of disrupting things, and I think that we should attempt, parallel to the research, towards the writing our own textbooks for our own consumption, because we are been reading textbooks written by the Western author, which describe their disease pattern, leaving out our disease pattern, and describing our disease pattern as case reports and something, you know, wondrous. So I think that is something that perhaps should begin to take discussion in this group. With that effect, with that said, I'm actually kind of foolish enough to have already embarked on writing a book on radiology for medical imaging for the low and middle income countries, describing their own disease pattern and how to utilize the available resources for the betterment of the patient, instead of following the Western protocols of studying patients in ultrasound, MR, and CT, so that all the resources are spent in a small number of cutting edge centers, and the budget is gone, and so nothing is left for remaining 80% of the population. I know that this was not the topic today, but just sowing a little seed into the minds of the greatest minds of the current time in Pakistan present on this forum. Thank you very much.
So nice of you, Professor Sarwat Hussain. We have just a couple of very open-ended questions. So there is a question, is there any study going on in Pakistan related to the after effects of COVID-19 in the COVID-19 infected patient after the pandemic? So I think that's quite a broad, open-ended. Maybe we can circulate in the group and we can find some right answer to this question. And the next one is Apna is having collaboration with very much all other medical institute. It should also have collaboration. So I think that's an invitation to collaborate with the med school. And then can't we bring all the trial unit to this group where we would enable us to drive research activity? So I think that was the missing link, that there was no platform available, a neutral platform. And generally in our culture, we are quite reluctant to share very openly. But I see there is a group, WhatsApp group, and there is a lot of activity going on in that group. And people are extremely excited, receptive, participative, and collaborative in that group. So I think that's quite a lot. And I must say thank you to Professor Samina, their team, Dr. Atif, and Muneen Akhtar, Saima Ahsan, Ghazala Ruby, and Dr. Umar Hayat, Dr. Sarwat Hussain. And with this, I hand it over to Dr. Sarfraz to say a few words and formally close the session with formal thanks. Over to you, Dr. Sarfraz.
Thank you so much. I know we are way over our time. So first of all, thank you everyone for the panelists for their kind words and their words of wisdom, and for the presenters for sharing their knowledge and expertise. And most importantly, thanks to all the participants for being engaged and joining. And we will announce the next in series very soon. We have our technical support with Waleed and his team, and we will be circulating the next webinar information soon. So thank you so much again, and wish you all very well. And we'll see you same place, same time, hopefully next week. Okay. Bye.
Thank you so much, Dr. Sarfraz, Dr. Uzair, and everybody who joined. Thank you. Allah Hafiz.
Thank you. All the best.
Allah
Thank you,
Hafiz.
Allah Hafiz.
Allah Hafiz.
Thank you.